Christie, and I'm Deputy Director here at the Isaac Newton Institute. And I'm delighted to be opening this workshop on active soft matter, the first workshop on the current 4-month programme on complex fluids. In fact, this is a pretty exciting week at the Institute, as we're also hosting the 4th workshop on the liquid crystals programme. If any of you think that you're meant to be listening to the liquid crystals talk right now, I will tell you where you should be in a minute. That workshop will be on liquid crystal defects and on active and solid liquid crystals. So we strongly recommend that participants on each workshop take a look at the programme and the list of attendees for the other workshop, as there are likely to be many items that you might find of interest. And if you're wanting to attend seminars for the other workshop, it's in Seminar Room 2 in the core building in CMS. That means you go out of this exit here of I&I, walk along up this pathway to the main central building, and then you go through the front doors and then just straight down the stairs there, and hopefully we'll have some signs up saying Seminar Room 2. And one of the good things is that it's been arranged that you are sharing your conference dinner with them, so that you should be able to have the opportunity to interact with them there. One thing that we really ask of you for this week, though, is that we are going to have up to 250 participants across the 2 workshops, and there's a summer school as well on one of the days. So can we please ask that you are really strict about keeping to timings, because if 250 people congregate for coffee at the same time, we're going to be in trouble. And on which note, please be patient if you have to queue slightly longer for coffee than you'd normally expect to. Details about meals and other practicalities should have been sent to you via email and are also available online. A couple of other practicalities: if you brought your car, it should be parked in the car park there, and you should get a notice from reception. If you hear a fire alarm, congregate in the aforementioned car park just over there. And other than that, I'm going to be so brief because you need to really be sticking to your time today. I hope you have a wonderful and stimulating week here for those of you who are visiting for the first time. And do remember to talk to the others, and we'll have signs over for CMS. So over to Alex.
Thank you. My name is Alex Lishansky, and on behalf of the organizing committee that includes, besides me, Len Disman and Carsten Kruse, I welcome all of you here. I think we put together a very interesting and diverse program, and it's my pleasure to open this morning's session with a talk by Raymond Goldstein from University of Cambridge. Please.
Thank you very much. Let's see. Is this on? Can you hear me? Good. Oh, yes. So I have 2 things that I want to make sure to say. First of all, I think we have to thank the organizers for gathering everybody and anybody who's in the field of active matter. And I just hope that nobody has the flu, because if we do, this whole field is going to collapse for a few weeks. The second thing is, if I look like I'm rather stiff today, it's because I'm recovering from a back injury, but I'm okay, so don't worry that I'm about to collapse. Okay, so about 8 years ago, I was visiting the University of Washington, and I was introduced to a biologist named Dina Mandoli, who told me all about a phenomenon which I only knew by name, which is cytoplasmic streaming. I knew absolutely nothing about it, but I came away from that discussion thinking this is a really interesting subject, and maybe physicists and mathematicians and fluid dynamicists could say something useful about it. So my purpose today is just to sort of summarize for you how we've been trying to understand some aspects of this phenomenon over the last 6 or 8 years. A lot of what I'm going to describe is basically experimental work done about 100 meters, 50 meters away in my labs. And during the week, if any of you want to come by and see the labs, you're welcome to do that. So the starting point is just to tell you that cytoplasmic streaming is the persistent circulation of the fluid contents of large eukaryotic cells. And perhaps the best place to see it is in large aquatic plants, such as this beautiful organism called Cara corallina, which grows in fresh water. It's about this tall, and it has individual cells that can be 10 centimeters long. 10 centimeters long. And it turns out that they have inside of them an apparatus which allows for the persistent circulation of that fluid contents in a very beautiful and regular fashion, and is trying to understand not only the microscopic physics of how that streaming is produced, but also what on earth it has to do with the organism that I want to focus on. Now, streaming was discovered, pretty much, everyone agrees, by one Bonaventura Corti, who wrote a fantastic little monograph in 1774, published in Lucca, Italy, in which he reported the discovery of this circulation using microscopes that were given to him by his wealthy patron. I wish I had a wealthy patron like he did. And in looking at Cara, which you see here, this is the kind of root-like structure that anchors it in the mud. And here are these so-called internodal cells that can be so long as I described before, and then interrupted by branchlets and more of these. And when he looked inside these cells, he saw what he drew as a kind of ring-like circulation in one cell and the next and the next and the next. And in fact, he used, his own urine to osmotically shock the cells and contract the inner vacuole and in order to see that actually there was this kind of strange structure on the inside with some kind of bag enclosing it. Now, here's a modern movie of the cytoplasmic streaming in Cara corallina. So the internodal cells are about 1 millimeter across and anywhere up to 10 centimeters long. What you see here is we're looking through an inverted microscope from below at one of these cells. These are all sorts of vesicles inside the organism that have settled to the bottom and are being carried along by this streaming. You'll note there's a diagonal line here defining the boundary between regions that move this way and regions that move this way. All the little green things are chloroplasts. And the speed of motion is something like 100 microns per second. And that actually means there are molecular motors that walk at 100 microns per second, which is probably a lot faster than most of you imagined. It certainly was in my case. The little things crawling around here are not part of the story. They are bit players hired for the day. Okay, so now cytoplasmic streaming is everywhere in biology, and there was a time in which people viewed its occurrence as essentially the definition of life. If you saw all this motion inside a cell, it must be alive. And so basically it's the circulation of the cytoplasm driven by the action of motor proteins, myosin along actin or kinesin along microtubules that entrain fluid and produce the large-scale circulation. So in aquatic plants and in some terrestrial plants, you can find it in the form of what's called rotational streaming, which is what I just showed you, and I'll explain the origin of the terminology shortly. In terrestrial plants, pollen tubes, for instance, you can find a kind of simple circulation streaming. In large protozoa, you find something which could be called streaming, but it's much more disordered and kind of bidirectional and not particularly regular geometrically. Now, one place where it's been discussed a lot in the past and in the present is in various aspects of morphogenesis. So, for instance, in the development of the egg cells in Drosophila, one can see on the scale of about 100 microns or so very vigorous streaming, which is driven by kinesin walking along microtubules and entraining fluid. It's very disordered. In fact, if you don't look at it this way, where you're seeing basically just yolk granules, but you actually stain the microtubules in green, you can see that this meshwork of microtubules is pushed around by the action of all these motor proteins in a kind of self-consistent way. They are the tracks along which the filaments move, and then they get pushed around in a very complicated way. So there are lots of interesting nonlinear dynamics in this problem of streaming, and I'm going to try to just pick apart various aspects of it as we go. So the history of it is rather amusing. So it was discovered by Corti in 1774, and then it was forgotten. And then it was rediscovered in 1811, and then it was forgotten. And this went on several times until the present era, when in 1956, Kamiya and Kuroda first really did the crucial experiments that pointed out that it's the sliding of the cytoplasm driven by motors which is responsible for the large-scale circulation seen inside the cell. And since that time, there's an awful lot of biological work on the nature of those motors and all of that, but a whole bunch of questions have been left unanswered. I'm sure you're all familiar with the famous essay by Haldane on the importance of being the right size. Are you? Have you read it? Please read it. I'll put it on my website. You can read it. It's a great essay, after dinner talk, I think, that he gave. And one of the things he said, which we all know is pretty obvious, is that beyond a sort of millimeter timescale, diffusion is not very effective in terms of transport, right? The diffusion time for 10 centimeters is 3 months. So obviously, if an organism has a cell that large, something else has to be giving rise to the transport. And a series of authors, Sochachka and Picard, over the years have pointed out that one possibility of this large-scale circulation is not just transport from A to B, especially in a closed cell where it goes A, B, and back to A, is some kind of mixing, some implications for homeostasis. And Picard, among all others, has, I think, most eloquently stated the questions that we should try to answer. Can we actually visualize the things that are being transported on this flow? Can we actually list them? I mean, even that isn't even accomplished. How do things know when to get on and off this flow if they are to go from A to B? And more importantly, for large plants which have these concatenated cells, there are tiny little nanometric pores called plasmodesmata that connect them together, and there might be a role for cytoplasmic streaming to carry things from one cell to the next. So there are lots of unanswered questions, and I'm going to tell you first how we use Cara corallina, this workhorse organism, to get at some of these questions. So here's the architecture. Again, many tens of centimeters tall plant, internodal cell a millimeter across. If you look very carefully at higher magnification, you'll see that this diagonal white line is actually a missing row of chloroplasts. So this organism has chloroplasts fixed on the inside wall of these cylindrical cells. They don't move at all. And here's a missing row. And if you take a cross-section, what you see is that there's a region of about 10 or 20 microns worth of the viscous cytoplasm, which is the metabolic goo of the cell. Then there's a vacuolar membrane, also called the tonoplast. And then 99% of the volume of the cell is taken up in the vacuole, which is this watery reservoir of nutrients. Now, if you look a little more closely, what you realize is that sitting parallel to these rows of chloroplasts are bundled actin filaments. And they're arranged in a very polar way. That is, on half of this cylindrical cell, on one hemicylinder, they are oriented one way, and on the other one, they're oriented the other way, so that the streaming is bidirectional, like a barber pole. And we now know from detailed studies of myosin 11, which is the relevant motor molecule, that the individual motors can move at speeds approaching 100 microns per second, which is at least an order of magnitude, if not two, faster than what we're usually used to for, say, kinesins on microtubules. Okay, so if you look closely near the indifferent zone, as it's called, that's the historical terminology, what you'll see is a beautiful regular flow, various things inside the cell acting as tracers, beautiful regular flow driven by presumably millions or billions or trillions of these motor molecules whizzing along on these tracks. There's no hint of the granularity. It's a very smooth flow. And if you look in a nodal cell, which is at the junction of all those branchlets, you see this beautiful, fast streaming just going round and round all day, every day. And again, the simple question is, what on earth is the purpose of this? So I'm going to try to answer some questions about what is the fluid velocity field in the cell? How do we relate it to the motor proteins? Does it have any implications for mixing? And what about this membrane, which bounds the cytoplasm and the rest of the cell? I don't have to tell this audience the importance of two relevant numbers in this world. Excuse me. First is, we're working in a low Reynolds number environment where there's absolutely no importance of inertia. But if we're talking about the transport of chemical messengers, which have a diffusion constant d, and we have fluid velocities u and a length scale L that could be measured in tens or hundreds or thousands of microns, then the relevant measure of the importance of diffusion to advection is the Peclet number. And the Peclet number, unlike the Reynolds number in these problems, can approach 1 or greatly exceed 1, depending upon the scale of sizes. So if we're talking about something like simple small organisms, bacteria, algae, that are 10 microns across and swimming at tens of microns a second, their Reynolds number is 10 to the minus 4, but their Peclet number is also sub 1. So they live in a world where diffusion dominates advection. But by the time you get to large multicellular organisms like this several hundred microns across Volvox or a millimeter across Cara cell, and we're talking about velocities of 100 microns a second at length scales of 1,000 microns, this Peclet number can be huge. So actually, these organisms do live in a world where advection dominates diffusion, and that would make Haldane happy because that was his whole point. So we do have to think about transport in a little bit different way in these problems. Now, here's a nice little problem for the student. I have a pipe, which is my Cara cell, and on one half of it, I have forcing at, let's say, a fixed velocity. Never mind the details of force generation. Just imagine a fixed velocity sliding one way, and on the other half sliding another way. Can you find the velocity field everywhere? I guess our Cambridge undergraduates can do that in their sleep. I have a little difficulty, but Picard worked it out in 1972, and here it is as a function of the radial coordinate and the angle. And this has the feature color-coded here of these two indifferent zones where the velocity field changes sign, and they're a very large amount of shear. So this is the basic structure of what we're after. Now, the question is, in Cara, is this really the velocity field? And one of the things that we realized early on was that because in Cara, this indifference zone actually spirals around the cell. It's actually bidirectional helical forcing. This broken symmetry makes for an interesting fluid dynamical effect. And you can see it here. If I if I put arrows to represent the forcing of these molecular motors, and I just imagine the projection of those two velocity fields on the horizontal plane, near this indifference zone, I see that they diverge from each other. And if I were to go on the other side of the cell and look at that indifference zone, I'd see that they converge. And that's a fancy way of saying that this object is chiral. It's not its mirror image. The two indifferent zones are not equivalent. And if you actually solve the fluid mechanics of a pipe with bidirectional helical forcing, you discover that in the cross-sectional plane of the pipe, there are two vortical structures that are produced. Now, these are reminiscent of what are known as Dean vortices that occur probably in your aorta. Whenever you have an inertial flow going around a curved geometry, you can get this kind of structure. But this is at 0 Reynolds number. And because the Peclet number of the overall flow can be so large, even the Peclet number of this transverse flow can exceed 1. So this makes for interesting possible implications for mixing in the system. And I'm not going to dwell on it in great detail, but I'll just show you a simple illustration of what happens. So suppose I imagine a diffusive flux of some scalar quantity into this cylindrical cell from the outside, and I had none of this mixing, none of this flow, none of this cross flow. Well, over time, the concentration field would just fill up to the interior by concentric diffusion. But as soon as I have this cross flow, I end up sending a tongue of this concentration field across, and that actually ends up mixing the contents. And in fact, for the realistic geometry of Cara, if you watch a little fluid parcel as it moves longitudinally down the cell, it also executes a kind of epicycle-like motion, and by the time you've gone one helical wavelength, it ends up on the other side. So I like to say this is the invention of what in some fields is known as the herringbone micromixer that came out of Whiteside's group in Harvard, but it's only 500 million years before. So nature figured out that by breaking this symmetry, one could actually get interesting kinds of mixing trajectories. They're not chaotic, but they are very nicely mixing. Okay, so this raises an interesting question. I've described to you a simple concept for this cell. It's a cylindrical tube. It has, let's say, some specified forces or specified velocities along the boundary. And this gives rise to large-scale flow. But remember, I told you there's a membrane. There's this vacuolar membrane that encloses the inner bag of water. And when I first learned that there was that membrane, that's how naive I was, I thought, oh no, this is terrible, because we know that the fluid velocity permeates the entire cell. We know that from early studies of the system. But if that membrane is sitting there, how could that be? It seems like it would block the shear flow. So one of the things we decided to do was to use the technique of magnetic resonance velocimetry through a collaboration with Lynn Gladden, who is a chemical engineer and our pro-vice chancellor for research here, because we learned through a workshop here, actually, that this technique, if one has a very steady flow, can have resolutions of a few microns or tens of microns, and therefore it would be possible to actually measure the entire flow field in this cell in real time and see if it conforms to this simple picture. So here's a Cara cell nicely put in one of these RF coils, and when you do the measurements, you actually are in a sort of Gaussianly enveloped way sensitive to a particular region of the cell. And here's the cross-sectional velocity profile obtained after several hours of averaging. And here another one and another one. And you can see the indifference zone spiraling here. So we get these individual pictures of the flow field. And of course, they're just rotated by the helical structure of the cell. And we can average them all together. Hello. And here's the experimentally measured cross-sectional velocity field going from something like plus 60 to minus 60 microns per second in this cell. And it turns out to look essentially identical to that theoretical calculation that just basically has sliding walls on a helical geometry. So all of this is to say not that we're such great fluid dynamicists, but that nature doesn't care about this membrane that's sitting 10 microns inside here. It's basically apparently just transmitting the shear as if it's not there. And this is something Picard had brought up a long time ago. How could this possibly be? I mean, if you think about it, a Cara cell that's 10 centimeters long has a lipid vesicle that's 10 centimeters long and a millimeter across inside it. And it's getting sheared, apparently, all day, every day, and surviving. So this led to a little question about exactly what is going on with this tonoplast. And to get a little insight into that, we learned a trick from Nina Allen, who's a very distinguished plant scientist who wrote one of the very famous review articles on cytoplasmic streaming. And she pointed out that it's possible if you expose a region of the cell to a very bright light for a few minutes, it will, over time, lead to the loss of the chloroplast, the detachment of the chloroplast there. They'll just float away, and you'll have literally a window into the cell. But otherwise, it's perfectly happy. And so if you do this, it's what you get. So here's a carousel, a millimeter across. Here's the window we created. And you can see that the streaming continues going. And if you look very closely, here's the remnant of that indifferent zone, and everybody's happily moving along. And so this is the start of trying to understand exactly what this vacuolar membrane is doing, because you can actually, in a sense, see it. You're looking straight on it here. But if you look from the side, as Vasily Kanzler and my group did, so you've got this circular hole and you just kind of look down like this, you can actually see this membrane getting pulled along by the cytoplasmic streaming. So here's the cytoplasm, and here's the vacuolar fluid, and it's just happily getting yanked along at that crazy speed of 60 or 100 microns per second. So I'm telling this kind of like a mystery story because that's how we went about it. I hope that's not distracting you too much. That's a little bit image processing remnant of some of the chloroplasts that are around. So it's not actually that shape. Yeah. Okay, so in thinking about this and trying to understand the role of the vacuolar membrane, we came across a very beautiful paper by Vezi and collaborators and Annie Violat's group in Marseille in 2007, in which they did the following very simple experiment. They took a lipid vesicle and managed to stick it to a substrate with some chemical glue and to put it in a flow chamber such that they could just flow fluid past it. Basically a simple shear flow, zero velocity here, constant gradient far away, and they observed, not surprisingly perhaps, the formation of this pair of vortices in the membrane, which they could visualize by little imperfections in the membrane that get carried around. Now, if you think about it, this is really very different than what you'd expect if this was just a drop of immiscible fluid in another fluid. Because there you could just push the fluid over and have it recirculate back in the bulk. And there'd be no problem with that because the only incompressibility condition that you care about is globally. But this lipid membrane is a two-dimensionally incompressible object. It can't flow back into the interior. So whatever flow field occurs on the surface of this must be two-dimensionally incompressible. And that's true no matter what the viscosity of the membrane. So this seemed like an interesting problem where we could just learn a little bit more about the nature of shear transmission and maybe even something about membrane viscosities. So my wonderful student Francis, who's around right back there, took up the challenge of doing this hardcore fluid mechanics calculation. I think that's the proper way to describe it. In all generality, you have a sphere that actually could be more than a hemisphere by some height h. It sits at a no-slip plane. You have some membrane viscosity with the shear force balance conditions on the surface. You have an imposed shear, and you want to find the flow everywhere. Now, excuse me. There really are only two important parameters in the problem. The membrane viscosity, as you may know, has different units than the three-dimensional viscosity because we're talking about a two-dimensional object. And so if I normalize it by the radius of the sphere and the viscosities inside and out, these are two dimensionless Safran-Delbruck lengths, which turn out to be the important parameters of the problem. And without going into any detail, the essence of this calculation is a series of modes, essentially, that are loosely speaking kind of like spherical harmonics that you can use to build up the actual flow field that you find in this vesicle. So indeed, you find these vortical structures inside the membrane. You can calculate the entire flow field inside the vesicle and start to ask questions about whether this is what we see in experiment. And so Aurelia Hunter-Camp-Smith, who's way back there with the Superman shirt, took up the challenge also of trying to do an experiment, to do a controlled version of the Violat experiment. So it's a microfluidic geometry in which you have a hemispherical vesicle, again a shear flow, and with some confocal imaging you can actually utilize membranes that are multi-component and display phase-separated domains which act as little tracers for the flow. And you can watch them move along under the action of the shear, and with different membrane compositions you have different membrane viscosities, and you can see the relative higher viscosity here where the motion is restricted under the flow and the very low viscosity here where the motion is very advected. And in principle, we thought, let's try to measure the velocity field everywhere and see if we can make sense of what goes on. So here are spinning disk confocal slices of tracer particles that are outside, above the membrane. The membrane's been dyed with a fluorescent dye, and there are microspheres inside. And at these different slices, you can see outer flow here, backflow inside. And in principle, with a fair amount of PIV analysis, you can actually reconstruct the flow field everywhere. And it looks exactly like what theory predicts. You have these beautiful, strange-shaped, closed streamlines inside the membrane. And you can also calculate and measure flow fields outside these vesicles. You can see closed streamlines even outside the vesicle. And then, in principle, by looking, for instance, at the attenuation of the velocity at the peak relative to what it would be without the vesicle, you can actually measure the membrane viscosity. And this is a rather heroic experiment. Theory predicts that if I look at the ratio of the velocity at the peak of the vesicle to what it would be in the absence of the membrane, r times the shear rate, as a function of 1 over the radius of the vesicle, I should get a straight line whose slope is related to the membrane viscosity. When you use a very low viscosity membrane composition, you discover that you basically see very much a zero slope, but an intercept that is exactly what is predicted by experiment. And at a higher ordered composition, you find a viscosity which is bang on what has been measured by other techniques where you look at rotational and translational diffusion of small domains in the membrane. So this gives us an idea, actually, that these membranes are in the regime of viscosity that's so low, actually, that they literally just directly transmit the shear. And that's a justification for what we saw in the experiments. Okay, so now I'm going to segue into some issues about active matter more by trying to ask questions about the patterns that are formed in these systems. So I've set up a kind of discussion of what goes on in these particular cells. But if we go back to Picard's calculation of the cross-sectional velocity, and we think about the rate of energy dissipation in the system, it's basically the viscosity times the square of the gradients of velocity in the system. We could ask, where is the dissipation coming from, primarily, in this bidirectional helical forcing? And if this is the cross-section, and here's one indifferent zone, and here's the other, this color coding tells you the obvious thing. The dissipation is where there's the huge shear. And so you might imagine, this is after the fact, I'll tell you, but you might imagine that if I had a soup of actin and myosin moving along, doing their thing, that they might be able in some way to self-organize to produce these kinds of patterns. And amazingly, there was a fantastic experiment by Yatsu Yanagi in the journal I'm sure you all read, Cytologia, especially that issue, in which he extracted from Kara droplets of cytoplasm, put them on a surface, and they spontaneously rotated. And so this is an amazing result, known to almost nobody, but it seems to show that a collection of motors and filaments and ATP, et cetera, will spontaneously organize, and maybe something like that is underlying the structures that we see in experiments. So let's try to think about this a little more. I sometimes say that this is one place where you could wheel out a mathematical theorem, because if I think of the inside of the cell like a Kara cell, it's basically got the topology of a sphere. And if I've got a vector field of actin-myosin forcing on it, obviously we know from the Poincaré-Hopf theorem, I can't avoid having singularities of some sort. I must have centers or sources or sinks. And in fact, if you look at the different types of streaming that are known in nature, you can see that they are just realizations of these singularities. So for instance, there's something known as fountain streaming, where you see this in pollen tubes, where the forcing is everywhere in one direction on the outside of the cell, or the inside of the outer wall, going up, and then it comes back through the center of the cell. And so there's a source right there. Or actually, sorry, that would be a sink. And the other possibility is that I go up here, and I force down here, and I have a source. And if you think about Kara, I've got these two indifferent zones. They're spiraling, yes, but if I straighten them out, and I shrink those lines to points, all I have is circulation. So maybe there's some sense in which this self-organization is trying to minimize dissipation or be most efficient. So we decided to try to recreate Yatsu Yanagi's experiment. So this is a an extract of cytoplasm of Cara that's been squeezed out, and you see it's very large, about 85 microns across. And when you first look at it under the microscope, this is sped up by a factor of 3, there's some weird motion going on, but nothing large-scale and organized. But you wait a few minutes, and that happens. Right? So Yatsu Yanagi was correct. There's some kind of self-organization into large-scale circulation. And if you try to do PIV on this, you kind of get the sense, that there is a simplification over time to something that is just a single vortex. So this led us to the following question. If we think of this as a bunch of motors walking along filaments, let's say they land somewhere on the filament, walk carrying cargo, because they're usually carrying vesicles of some sort, and then detach, it's not hard to see that the net effect of that motion is essentially like a stresslet in fluid mechanics, because you have the motor going one way, you have the filament going the other way. That's a forced dipole. And statistically, as long as the motor stays on the filament for a fair length of time, it's going to be motor closer to one end than the other, and that's a pusher dipole, like a bacterial cell. And so using the technology that has been developed by much of this community actually over the last bunch of years, it's possible to build a statistical theory of the possibility of pattern formation in a system that it consists of these filaments, but enclosed in some kind of box. Let's call it a circle. So this is something that Francis worked out, and it happened to coincide nearly identically in time with the beautiful work of Zvonimir Djodjic's group, which I'm sure we'll hear about later this month, where things like this and many other beautiful things were seen. So the result of this calculation, I'll just summarize, is that there's a critical stresslet strength, product of the typical forces of those dipoles and the separation between them, for the onset of spontaneous circulation in a confined geometry. So it's related to the fluid viscosity, the concentration of those dipoles, and the two diffusion constants, rotational and translational, assumed for the elementary objects. And when it forms this kind of spontaneously rotating state, it turns out that there's a kind of spiral arrangement of the individual filaments. Now, we're working on trying to do this in a controlled way with something like Cara, but the fact that elementarily what's going on here is forced dipoles led us to wonder whether a similar kind of thing might occur if we used a different kind of forced dipole, a self-propelled one, namely bacteria. So this led to the idea of using the same kinds of organisms that we know give rise to these wonderful turbulent bacterial collective dynamics, namely Bacillus subtilis or E. coli, in these kinds of confined geometries. So I'm sure many of you have seen this movie of collective behavior in Bacillus subtilis on the scale of several hundred microns. This is actually in a petri dish near the three-phase contact line. Here are microspheres being pushed around to show you that the fluid also gets jostled around. So what would happen if you put this in a confined geometry? And so Hugo Wieland, who should show up here momentarily from Paris, there he is, right? Decided to have a go at this. And he developed a beautifully clever experiment in which he made an emulsion of a bacterial suspension. So you make a bacterial suspension, you basically add some surfactant and mix it with mineral oil, and you end up with droplets of this bacterial suspension stabilized, and they can be then squished between cover slips, and you have a kind of pancake-shaped bacterial suspension surrounded by a higher viscosity medium. And actually, today, this paper will come out, I hope. So what do you see? Well, if you have a very large droplet, this is nearly 100 microns across, you see just a snapshot of that collective behavior I showed you before. So it's just confined in a circle, but no big deal. With some image processing, you can actually start to kind of see the cellular alignment here. But you'll agree with me that this is not a highly ordered system. But if you make smaller drops, what you see is a beautiful spiral vortex. And you can see the kind of spiral behavior of the individual cells here. And there's something weird going on here. You're thinking that maybe there's an optical illusion, but no. The cells right near the edge are going the opposite way. And this is really strange. And in fact, you can look at this using PIV to track not only the individual cells, but also the microspheres that you may add. The velocity field has the feature of going to 0 in the middle, peaking up and then coming back down, and actually going negative right near the edge on a length scale that is pretty constant with drop size, basically 4 microns. So that backflow is basically one cell thick and independent of size. And it turns out that if you construct an order parameter that basically just measures how well organized this flow field is in the azimuthal direction, as you change the drop diameter from, say, 20 to 100 microns, there's a window inside of which you'll get this nice, highly ordered vortex. And if you're too large or too small, you don't get it. But what's going on inside is really strange. And we've been trying to figure out exactly what's going on. But it looks something like this. The individual cells at the edge of the drop appear to prefer to parallel park at an angle. And you can kind of understand this basically in terms of a certain volume fraction in the system and the need to pack it. Obviously, you could tile this surface with a bunch of guys lying flat or lying perpendicular, and somewhere in between is the angle associated with a given volume fraction. But as they are pushing fluid behind them with their flagella, this backwash creates collectively a kind of shear flow. Since the fluid outside is higher viscosity, it's a kind of shear flow with a stick boundary condition here, and this will tend to push them over in a self-consistent way. But then that flow field pushes this way and will make the next layer of cells go the other way. So this is a hand-waving, very hand-waving argument as to why there is this backflow. And if you look at the angle that the cells make with the boundary as a function of drop diameter, and just construct some very simple geometric bounds on this, given the size of the cells, you find that it's pretty consistent with the experiment. But what it shows is that if you want to make a continuum model of what's going on here, you have to be very careful, because on a micro scale, something very cellular is happening that has to do with the way that they actually organize.
But what is the orientation? If it's a spherical group,
Oh, gosh.
some angular.
I don't know. Well, there'll be a singularity somewhere. We haven't done that experiment yet. Okay, now the other thing you can do is ask what happens if the volume fraction is less than the very high value that we used, which was approaching something like 30 or 40%, to get that earlier picture. And what you see is that the cells migrate to the outside. Essentially, they have inelastic collisions with all their other fellows, and they end up stuck on the outside, clearing out the center. And so they achieve this concentrated boundary layer independent of the fact that the overall volume fraction is less than maximum. Okay, now, just going to segue out of this for a moment. Kayla Lushi, who's also here, has visited us for a few months as a Crichton visiting fellow. And just in the last couple of days, she's begun to do some simulations. If they'll play. Oh, come on. It's not playing. That's not good. She's begun to do some simulations to show exactly this. This will teach me for trying this. Let me try again.
Try shutting it, opening it, and
Yeah,
opening
probably.
it again.
That'll teach me. There we go. Sped up, just for your amusement. So a bunch of cells which are not allowed to cross each other. They have hardcore repulsion, but they have true fluid mechanics going on in terms of the flow fields created by flagella. And what you see is that they spontaneously organize to clear out the center and to produce this kind of vortical flow, and they align in this chiral spiral way. So it seems that those are the key ingredients, basically hardcore repulsion, pusher-like dipole structure, and that gets you all of this. Okay, so you can do lots of interesting things with this fluid, which is a kind of new kind of fluid that is this concentrated bacterial suspension. For instance, you can ask, what happens if I have a structure that allows or coerces two different vortices to exist, and how do they talk to each other? And what you discover is that this little coupling in between naturally tends to make them rotate in opposite directions, as you might imagine. So then you can start to play all sorts of games of maybe making a triangular lattice of these or a square lattice of these and asking about what kinds of large-scale structure appears. But what's going on is really very interesting because there's this strange boundary layer of cells doing its thing, and then it's coercing the rest of the structure on the large scale. So it's very, I think, interesting to ask what kind of continuum model should I use to describe this kind of behavior? Maybe I just have to muck up the boundary conditions the right way. But it's a very complicated, somewhat non-local problem. Okay, so the last couple of minutes, I just want to give you a little bit of a flavor for how some of these ideas could be used to take us back to the original problem I started with, which was this beautiful aquatic organism, Kara, and its very regular geometry of streaming, to see if we might find in there some kind of self-organization process. Now, perhaps the most important experiment that was done in this field was done back in 2000 in another journal, which I'm sure you read a lot, Protoplasma. There's some very interesting journals we all have to start reading these days. And this was an experiment done on Kara in which various chemical interventions were used to disrupt streaming. Now, cytochalasin D is known to stop streaming, and oryzolin is something which is known to disrupt microtubules. Now, interestingly, even though I talked about this problem as if it's just myosins walking on actin, there are microtubules implicated as well. And these authors found that with the right concentrations of these two components, they could disrupt streaming completely in the cell and disrupt the actin mesh enough that when they allowed streaming to reestablish by removing these chemicals, the streaming would appear in a different geometry. So for instance, if these arrows represent where the original indifferent zone was before this experiment began, where the two areas of fluid went past each other in opposite directions, after the experiment it had moved to where you see in red. So the new indifferent zone is here. So schematically, basically, we have a situation like this. All of the underlying railroad tracks on which the motors move have been disrupted. They reform, and it moves in a new direction. So this strongly suggests, that might be for me. This strongly suggests some kind of self-organization process at work. So with Francis, we thought it would be good to try to build a phenomenology of this, and I'm just going to be super brief on this. There are a couple of important things that we think are operating here. Each by itself wouldn't be enough to make this happen. One is that when these motors are walking along the filaments, of course, if there's already a background flow from the other guys who might have organized, then it's one thing for a vesicle to be moved this way in the direction of the flow that's perfectly stable. But if a vesicle is moving in the wrong direction, there'll be a hydrodynamic force that tends to try to move it around. And that can be a force which rearranges the underlying filaments. Of course, given any intermediate arrangement here, there can also be a pair of forces acting on a filament vesicle complex, which will tend to align it. We also know that this underlying behavior does not occur unless the actin filaments are able to bundle. And finally, for various reasons also coming from the microtubule world, there's reason to believe that there's a coupling between the orientation preference of the filaments and the underlying geometry of the surface. Obviously, it's a lot easier to align this way than this way. And if you build a continuum model very much along the lines that we've all seen many times for these active matter systems, and put it on the geometry of a cylindrical object, what you discover is that the coarsening process that occurs over time will take a rather disordered initial configuration of filament orientation and flow and eventually turn it into a very simple stripe-like arrangement of motors and filaments walking in this bidirectional way. So this suggests to us avenues to investigate, to try to see further whether this hypothesis that self-organization is behind these kinds of geometries is true. It might not be, but we think there's some potential interest in this. Okay, so I'll stop there. I've told you of the work of an awful lot of people, starting with my former postdoc Idan Tuval, Vasily Kanzler, who's moving to Warwick this week, basically, Aurelia, who's at the back, and Jorn Dunkel is a theorist in the group, previously with Julia Yeomans. Jan Willem van de Meet started out all the work on Kara. Francis, Ugo, and Philip have worked on streaming in various ways. And I also just want to reiterate the very important collaboration with Lynn Glyden's group on the molecular, the magnetic resonance velocimetry. Thank you.
Questions?
I'll just repeat a little bit. What is the preference of rotation, clockwise, anti-clockwise, and spherical droplets? It should be orientation of angular velocity. How did do the system?
So statistically, there's no preference to go counterclockwise
So
or
in
clockwise.
experiments you see whatever with equal probability, yeah.
Yeah, I mean,
And in three
there's no Coriolis force we have to worry about.
in spherical droplet you have any orientation of.
Very hard to visualize that kind of geometry precisely, and so we don't have any real data on a truly spherical drop and what it does. I'd love to. That would be great.
But does it stay the same? It also can fluctuate, say in spherical droplet. It can orientation of angular velocity can
Yes, I'm
change
sure those
or
are.
not. Or
I'm
there
sure they're possible,
some
but
conservation of like angular momentum or whatever.
well, I think at some point we thought that there was an angular momentum argument for this backflow, but then we realized it's really just a physical argument about what the cells are doing near a higher viscosity interface. In fact, if you just put them in a PDMS chamber where there's infinite viscosity outside, they do exactly the same thing. So we think it's just a no-slip condition, effectively, at the edge that makes for the backflow. But we don't have any information on a full 3D drop.
Okay.
When you were describing the experiment with cytoplasm in a drop, you were using language stating that the flow is due to what's happening in the bulk. Do you know that's in the bulk as opposed to something going on the surface? Because you could imagine that as well.
We don't really know. We'd like to try to label what's inside in a way that would allow it to keep going and give us spatial resolution. We're not there yet. So we don't, in that particular experiment is not particularly controlled. It's anecdotal and motivational. And I agree with you completely. This question of whether it's at the edge or inside, I think there's some indication that you can get some very strange behavior at the edge. And you might imagine the filaments over time would plaster themselves against the wall. So it could be.
Ray, I have a question about this experiment, bacteria swimming in small droplet.
Yeah.
And in the model, you assume that hydrodynamic interaction is important. I'm pretty much sure if you just assume inelastic collisions and self-propulsion, we will have this pattern with a hole in the middle. And bacteria moving.
Could well be. I'm with you. I mean, I think one of the big questions that's been in this field for a long time is whether hydrodynamics really matters. I originally thought for sure it did, and now I pretty much think no, it doesn't.
No, I think
But it depends on what you mean by hydrodynamics in the sense that whether I really have to worry about large-scale algebraic fall-off flow fields at large distances, I think the answer is no. But if I have to worry about lubrication kind of dynamics, then maybe yes. But I agree with you that a lot of it is just steric. Yeah.
So Ray, what's the smallest, the simplest dynamics that would create rotation in a ring?
You mean from a biochemical point of view? Or?
No, but computational point of view, modeling point of view. What's the simplest thing you would need to have to get these guys to start turning around?
Well, I mean, the fact is, if you go back to something like this picture, you're basically, I think, saying there's a characteristic length scale in the problem, which is a kind of coherence length of this fluid. There may be some boundary conditions, but fine. So it's also generating its own internal energy, right? So you need a system that's non-variational, that has a length scale, and essentially picks out characteristic velocity. And one way to do that is essentially take the kind of toner 2 type phenomenological generalizations from long ago, and just basically look at a negative diffusion term and a fourth order stabilizing term, and it's kind of like a Swift-Hohenberg equation that's non-variational, and it basically produces a characteristic length scale, a characteristic velocity, and it will do many of the things that we see in collective dynamics, and I suspect it will also do something like this. So that's a phenomenology of how you might do it. It's basically, you have to have energy injected continuously, you have to have a length scale, and sort of tendency to form a characteristic velocity. Does that answer your question, or make it worse?
You don't particularly need a characteristic self-propulsion velocity because in a closed geometry, simply having force dipoles without a polarity is enough to produce circulating flows. The polarity is not crucial to produce.
No, you're right, absolutely.
So that's one point.
I guess what I was remarking in part because he said the simplest. So I mean, if you just ask what equation do I write down for the velocity field and nothing else, then that would be the answer. If I allow for a suspension dynamics where I have two different fields, then yes.
Just one short question. You imposed incompressibility on the surface of the vesicle.
Yes.
Isn't there any tendency to move into the third dimension, in which case?
For these small vesicles, they're sufficiently taut that apparently not. I mean, we have 3D images, and you can get to a regime where you distort them. It looks like a raindrop on your windshield, that's hit by the wind. But most of the time, they're very rigid. But there are visualizations of streaming dynamics in Arabidopsis, for instance, where there are proteins that are fluorescently labeled in the tonoplast, and you see dynamics that looks almost like vesiculation. So if it's floppy enough, as Corti managed to do, I think you'll get all sorts of really interesting dynamics.
To reach you 100 micron speed, I'm sorry, there is a ratio between the speed of the motor and the speed of the flow. And the speed of the flow should be always smaller than the speed of the motor. It seems that the two are very close to each other. Does it mean that there are lots of motors cooling at the same time?
So I think the answer is there are lots of motors. I mean, there's a scaling argument that goes back to Picard about how the measured fluid velocity would scale with the diameter of the cell as it grows. And you make various assumptions, but if you just have a fixed concentration of motors on the boundary, you get what's seen in an experiment. And that concentration is pretty high. I mean, we're talking about lots of motors per filament all the time working. However, there's an interesting idea from Dan Frankel's group in chemistry here, which I think merits a lot of study, which is whether there's something analogous to what you see in a bicycle race, but at low Reynolds number, namely grouping together that can increase the speed. I mean, you might imagine that given the typical force-velocity relationships for motors, if I am surrounded by all sorts of others that are producing a flow, then without any work, I'm already moving a certain velocity, and therefore if I consume ATP, maybe I can move faster than I normally would. Obviously, I can't do that forever, but there could be a speed-up.
Do you know the step size of the motor, or are they always bound, or are they always unbound?
I have forgotten the level of precessivity of this particular myosin. But we can, in principle, you could compare what you see in single molecule studies and this, and they are pretty much the same.
I suggest that we stop here and thank Wayne. It's a coffee break, Back at 11.